And I'd like to share with you a little bit about our work in computational drug discovery. But before I start, I always like to say why I'm excited to be a computational biologist today. And one of the reasons data that's publicly available that really enables us to ask exciting questions about disease. These are just some examples of these resources. The Gene Expression Omnibus, for instance, contains pretty much any microarray or any transcriptomic study that has been published or NIH funded in the last 20 years. The 1,000 Genomes is an example of a genetics data set that's often used as a reference. Import is a data repository, an immunological data repository that contains a lot of clinical trial data sets. We'll talk about the LINKS data set later in the presentation as well, which is looking at drug effects and expression effects that are induced by treating with certain compounds. And the cancer Genome Atlas, TCGA, is an incredible resource that the cancer community has put together. And I'm glad that a lot of these data sources look familiar. A lot of us use them in our everyday work. And the other reason for, why I'm excited to be a computational biologist now is that, the cost of sequencing has gone down tremendously in the last 20 years. And so has the cost of computation. So our phones that we have now are much, much more powerful than computers were only a few years ago. And that just provides an amazing opportunity. As a result, we can profile individuals and look at disease from a multi-omic perspective. So the genomics and transcriptomics have been around for a while, but now we can look at the antibody repertoire. We can study epigenetics. We can look at proteomics. We can look at the microbiome. And integrating all of these measurements is something that my laboratory is very interested in. And moreover, now you can generate many of these types of data on a single cell level. And that also provides both an opportunity to explore some of these important signals, but also is very interesting from an analytical perspective and provides a whole level of complexity on the analysis level. So in my lab, the types of questions that we're interested in is how can we use data integration to advance biomarker discovery? How do we identify better diagnostic markers? How do we improve therapeutics by finding new uses for existing drugs through drug repurposing? And how do we understand disease mechanism a little bit better through analysis? So today I'll focus on our drug discovery work. And the reason and the motivation behind drug repurposing is fairly simple. It's expensive and it takes a long time to take a drug and bring it to market. And 90% of drugs fail in early development. One solution to that is drug repurposing, which is the idea that you can take a drug that's already FDA approved and identify a new use for that compound that reduces the cost and also reduces the risk of failure. There are many different ways to do computational drug repurposing. There are target-based approaches which leverage genetics data. And there the idea is if a certain genetic variant or a certain gene that's associated with a disease, either through GWAS or familial studies, is associated with several diseases, then maybe a drug that targets that gene or protein can be used across different diseases. There's what I call disease-based drug repurposing. And there the idea is you might have diseases that manifest themselves similarly based on clinical data or molecular data. And for instance, that's one example of that is autoimmune disease. And maybe a drug from one of those diseases can be repurposed for another disease. There are molecular modeling approaches, docking-based, and kind of looking at the interaction of structure of drugs and their potential ligands. I'll be honest, I'm not an expert in that area at all, but there are many, many very smart people working on it. There are companies that are doing this. So it's a pretty wide area of research. And finally, there are these network global signature approaches where the idea that one drug, one target, one disease might not be sufficient for complex diseases. And this is really the type of approach that we've been pursuing. So the question that we've been interested in asking is, can we use public gene expression data to systematically predict relationships between drugs and diseases. And let's say we have gene expression data from disease, so healthy individuals versus diseased individuals, and we have gene expression data from treatment with different compounds. Can we link the drugs and diseases using gene expression as a proxy? So what we want to end up with is a matrix of diseases by drugs, where each cell of this matrix is some sort of a therapeutic score of how likely a given drug is to be efficacious for a certain disease. So what do we do first? We go out and look for data. So here we used on the disease side, data from the gene expression omnibus, and these statistics are a little bit, actually probably a lot out of date. I think GEO has over 4.5 million samples now, and the more impressive number is this number of series or number of experiments. I think that's over 100, definitely over 100,000, if not 150,000 different experiments, many of which, of course, deal with disease. The drug data that we've been leveraging comes from the connectivity map. And this is a resource developed by the Broad Institute, which is a collection of expression data from cultured human cells. So they took a bunch of cancer cell lines, put a number of different compounds. The initial data set was 164 compounds. The extended data set is about 1,000 drugs. And they measured gene expression before and after treatment. And this repository contains both FDA-approved drugs and non-drug bioactive small molecules. Our hypothesis was fairly simple. If the disease expression signature is reversed by the drug, then maybe that drug could be used as a therapeutic. So we developed a computational pipeline to take a disease expression signature and query it across the drug data. And we come up with a drug disease score. You can really think of the score as a correlation. So the positive relationship are profiles of drugs that are similar to the disease, and the negative relationships are profiles of drugs that reverse that disease signature. And that's what we're really interested in. This is a matrix summarizing the results of our initial investigation. We looked at about 100 diseases across 164 drugs, so the limited data set at first. And we were able to identify therapeutic candidates for about half of those diseases, for 56 of them. Here in this matrix, in green, you see the negative score, so the therapeutic predictions, and in red, you can see the positive correlations. So what we really wanted to do, though, looking broadly is interesting, but we really wanted to focus on a single disease and see if we can predict a therapeutic for it. So the disease that we chose to focus on was IBD and Crohn's disease. This is an inflammatory disease of the intestine that has an autoimmune component and affects about 500,000 people in North America. And there's no known pharmaceutical cure. The current solutions are to reduce inflammation with anti-inflammatory drugs and corticosteroids, which have pretty bad side effects and also just treat the symptoms. There are also surgical solutions that are quite invasive. So looking at our therapeutic predictions for Crohn's, we saw prednisolone was one of the top hits. However, there was one other drug called topiramate that had a better therapeutic score. So it turns out topiramate is an anti-seizure drug, and it suppresses the rapid and excessive firing of neurons at the beginning of a seizure. It does so by enhancing GABA activation, and it's been used to treat epilepsy and bipolar disorder. It's also an antidepressant and has been investigated as a potential treatment for obesity and type 2 diabetes. So we partnered with both some collaborators. This was work done at Stanford, and also 2 independent CROs to validate this prediction. So we had 2 follow-up validation studies. Well, actually 3. So 2 CROs and a collaborator. One of them, the biggest one, I'll describe here. There were 4 groups of 12 animals each, and we used a TNBS animal model for Crohn's disease. The 4 groups were healthy controls, the disease model induced that were untreated, disease model induced that were treated with prednisolone, and disease model induced that were treated with topiramate. The study went on for 7 days, and we collected a number of different types of data. We collected clinical science, pathology scores, histologies, and even endoscopy images on these animals. So this is what the data looks like. In terms of the clinical signs, this is percent of animals with diarrhea. In green, you see healthy controls who are doing great. In red, animals with a disease model induced that were untreated. And in blue and purple is topiramate and prednisolone treatment. So we saw the symptoms going down significantly with both treatments, the kind of gold standard that we used, as well as the predictive drug. However, when we look at the microscopic damage score and the gross pathology scores, we saw a significant reduction with topiramate that we did not see with prednisolone treatment, shown in these graphs here, both when we look at the gross pathology and the microscopic damage score. And these are the representative histology and endoscopy images for the animals. We then went back to the signature that we used for prediction, and we wanted to see what are the pathways that are involved. So you can see the disease signature on the right, and you can see that it's reversed by the compound. At least if you squint, it's not perfect, and there are many caveats to discuss here. But the pathways made quite a bit of sense. A lot of immune pathways, IL-15 signaling, B-cell receptor, epithelial cells, inflammatory response, antigen presentation. And when we looked at a handful of genes, we saw that, for instance, TRPV1 is a gene that was downregulated in the disease model and upregulated in the drug. And IFI30 was the other way around, upregulated in the disease model and then brought back down by the drug. So this was the first application of this approach. The method was developed actually 10 years ago, and this was part of my graduate work back at Stanford. But we've applied it to a number of different disease areas and adjusted it along the way. So IBD was the first application, but we've also looked at dermatomyositis. Cancers are really a logical application because the drugs are tested in cancer cell lines. So I've collaborated with several folks to look at basal cell carcinoma and also liver cancer. And we've looked at pregnancy outcomes, specifically preterm birth as another disease area. And most recently, of course, we've applied this to COVID-19 as well. But I'd like to spend a little bit of time talking about the application of this approach to neurodegeneration and specifically Alzheimer's disease. This is a review paper that we published a few years ago that you can kind of dive in deeper in terms of approaches to computational drug discovery and drug repurposing in Alzheimer's disease. And actually, earlier this month, we had a big paper come out doing kind of applying a precision medicine flavor of this approach to ApoE-related Alzheimer's disease specifically. So I'd like to tell you a little bit about this story. So this was work done in collaboration with Dr. Yedong Huang at the Gladstone Institute, and the lead author is Ali Tabs, who was the first graduate student in my lab. And of course, many, many other collaborators on this study. So here, what we really wanted to do is we wanted to apply a precision medicine approach to drug repurposing. So what does that mean? That means maybe Alzheimer's disease is not a single disease, but maybe it's many different types of disease. And here, what we wanted to do is we wanted to stratify the patients based on their ApoE genotype. You might know that ApoE is the biggest risk factor for Alzheimer's disease. So instead of aggregating all of the Alzheimer's disease patients in one group to generate the disease signature, we generated the disease signature separately for individuals with ApoE 3.3, 3.4, and 4.4 disease. So for each one of these, we generate a disease signature. And this is the data set that we used. You can look at the distribution of case and control samples across the genotypes. This is a PCA plot visualizing all the data. And you can see that there is a split between case control. These are brain samples from these patients. And you can also see that there is at least some structure with respect to genotype, where the 3.3 individuals are shown in blue, the 3.4 individuals are shown in gray, and the 4.4 individuals are shown in red. Looking at the genes and the pathways across these signatures, even before querying the drug data, you see that while there is an overlap across these signatures and pathways, there's quite a bit that's specific to the genotype, especially looking at ApoE 3.3 and ApoE34. While there's an overlap, there are some signals that are distinct. So then we continued to query the drug data for these three signatures separately. So this is what the results look like across 4-4 disease shown here, 3-4 disease, 3-3, and then everybody together. And we noticed that looking at ApoE44 associated Alzheimer's, we saw that bumeranide, which is a diuretic drug, was one of the top hits in our therapeutic predictions. It still has a negative score across the other comparisons, but it's strongest when we look at ApoE44 disease. This is just digging into the signature a little bit more. You can see here at the top, the signature for ApoE44 Alzheimer's versus the drug results. So turns out bumeranide is a loop diuretic that works by decreasing the reabsorption of sodium by the kidneys, and it's used to treat edema and hypertension. So it's pretty widely used in the clinic. The drug name is Bumex. So we partner with Dr. Huang's lab, and he has an ApoE44 animal model for Alzheimer's disease. So not only was our therapeutic prediction based on this precision medicine genotype-driven approach, but we could also validate in a similar way. So we started a whole set of animal studies, and I'll just show you one of them in the interest of time. This is a behavioral study where the animals are placed in a pool of water where there's a hidden platform, and they don't really like to be in the water, as I understand. And I'm a computational biologist, I'm not a wet lab biologist, but this is my understanding of how this experiment works. And the animals search for this hidden platform to be able to get out. And they try to remember where that platform is, and this experiment is repeated many times. So what we're looking at here is how much time do the animals spend in the target quadrant of interest versus somewhere else. So this is what we observed. The animals with ApoE44 genotype, here shown in red, that are untreated, spend a longer time in the other quadrants as opposed to individuals who were treated with bumeranide. So the dashed lines are individuals who are treated with bumeranide, and you can see that from the 4-4 individuals that are shown in red, there's a significant reduction. So basically, these individuals look very similar to the animals that don't have the disease deficits, so the 3-3 group. So this was our kind of initial validation. There's quite a bit of additional work looking at electrophysiology and also looking at the transcriptomic effects of bumeranide in these animals that confirm our predictions. What we're doing next is we're leveraging single cell approaches to move forward. So this is work by Stella Belan-Wu and Yachao Li in my group. It's under review, looking at whether we can leverage single cell data to understand ApoE44 disease a little bit better. I will skip this in the interest of time and show you a little bit of one step further that we went computationally with this bumeranide study. The question was, can we use real-world electronic medical record data to further validate these predictions? So here, we leveraged clinical data from UCSF to begin with. And the clinical data spans from 2012 to today. We have about 1.3 million patients, and we have all sorts of information, including allergies, diagnosis codes, lab test results, and medications for these patients. So given this data set, we wanted to see whether we can look at the patients that are on bumeranide already and see what is their prevalence of Alzheimer's disease. So we actually did this both at UCSF and also independently at Mount Sinai, and wanted to see what does the data look like. The work at UCSF was carried out by Edith Kosti and Tomiko Oskotsky, and the work at Mount Sinai was led by Mizut Bisak in Ben Glicksberg's lab. So looking at both the UCSF and the Mount Sinai data, we see that patients who were taking Bumeranide have a significantly lower prevalence of Alzheimer's disease, both at UCSF and at Mount Sinai. So this is an example of a study that started with molecular data where we did a targeted prediction focusing on ApoE44 disease. We validated it in a number of animal models, looking at behavioral studies, electrophysiology, the expression data that I didn't have a chance to talk about today. And then we went to human data in the electronic medical record, which also confirmed our hypothesis. Unfortunately, we did not have genotyping information on these individuals from the EMR, but nonetheless, even without that, we see the trends are fairly significant across two independent institutions. So now we're moving further in leveraging electronic medical record data to better understand disease heterogeneity and specifically sex differences in Alzheimer's disease. This is a visualization of all the Alzheimer's disease patients at UCSF on a dimensionality reduction plot. And you can see that there's, based on their comorbidities and ICD-10 codes, and you can see that there's a significant shift between patients with Alzheimer's in red versus healthy controls in blue. And I don't have time to talk about this in detail, but this is fantastic work by a very talented graduate student in my lab, Alice Tang. And the paper is available on medRxiv if you're interested. We're further looking into drugs using the electronic medical record data. So all of this is ongoing. So in summary, I told you about our computational drug discovery efforts. In the context of Alzheimer's, we established this ApoE genotype-dependent transcriptomic signatures from publicly available data. We identified Bumeranide as a potential therapeutic, and we saw that it rescued AD-like cognitive and electrophysiological deficits in mice. We're looking into single nucleus sequencing to explore effects post-treatment in mice and human cells. And finally, we used electronic medical record data to show the prevalence of Alzheimer's is associated inversely with Bumeranide treatment using real-world data. In terms of ongoing work, we're looking into combination approaches, applying machine learning, further leveraging single-cell data and electronic medical records, and investigating sex-specific differences in Alzheimer's disease. And my hope is that by integrating the molecular and the clinical data, we can get a little bit closer to the goal of precision medicine.